This video is going to cover the answers for the worksheet for chapter 11 and 12. First, we are going to be going over matching, which is in part A. We're going to have some terms and we're going to match them to their definitions. Let's get started. So the first one that we have is transcription. And then transcription is going to be the letter G, which is the process by which DNA is converted to RNA. Number 2, the Schindel-Garneau sequence is going to be the letter E, which is the ribosomal binding site that is found in bacteria and is located upstream to the start codon. Number 3 is going to be J, which means that is the nitrogenous base that is only found in RNA. Number 4, the Tata box, is the letter I, which is a eukaryotic region of DNA that helps initiate the process of transcription. Number 5, which is the Pribnow box, is going to be letter L, which means that it is a part of the promoter-sided DNA for transcription that occurs in bacteria. Number 6 is letter A, which is the enzyme that catalyzes the synthesis of RNA. Number 7 is C. The Rho factor is a prokaryotic protein involved in the termination of transcription. Number 8 is letter B. The coding strand is the DNA strand whose base sequence corresponds to the base sequence of the RNA transcript. Remember that in the RNA transcript, we're not going to have any T's. We're going to have U's instead. Number 9, which is the poly-A tail, corresponds to the letter H, which is a type of post-transcriptional modification. Number 10 is going to be letter D, which is the ribosome is a protein complex that is involved in RNA translation. Number 11, which is template strand, is letter F. That is the strand that is used by DNA polymerase or RNA polymerase to attach complementary bases during DNA replication or RNA transcription. And lastly, we have number 12, which is going to be letter K. An anticodon is going to be a sequence of 3 nucleotides in a transfer RNA molecule that corresponds to the complementary codon in messenger RNA. Let's move on to part B. On part B, we are going to have either given to us the mRNA, the DNA, or the amino acid sequence, and we have to fill the rest. Understand that even though it was not stated in the problem, I choose to write the coding strand and template strand for illustrating that DNA is always double-stranded. So, if you are given the mRNA, the easiest thing to do is to write the coding strand. Because in the coding strand is going to be pretty much the same sequence as the mRNA, it's just that the U's are going to be replaced by T's. So in problem 1, the coding strand is going to be a TG, a CT, a GC, TGG, GGG, TAT, TAC, TTT, and the last one is TAG. Okay? If now I write the template strand, and for the purposes of this course, understand that the template strand is always going to run 3' to 5'. The coding strand will be the one that runs 5 to 3. That's just for simplifying the whole process. When it comes to the template strand for this sequence is going to be specifically TAC, TGA, TCG, ACC, CCC, ATA, ATG, AAA, ATC. Okay? Now, the tRNA is going to be present in the anticodon, and this is going to be base pairing with the mRNA. So understand, because we are having, in essence, an RNA-RNA interaction there, then understand that the base pairing here is going to be A to U. Let me just write it bigger. G to C, C to G. So the tRNAs that are going to be bringing in those amino acids based on the first, on the information given on number 1 are the following: UAC, UGA, TCG, ACC, CCC, AUA, AUG, AAA, and please note that there is no tRNA for the last sequence, and I will explain in a few minutes why that is. Remember that when we are determining the sequence of amino acids, is determined by the sequences that we have in the mRNA. And I highlight, as you can see, in gray. So we are going to be paying attention to these 3 letters sequences that I underline in gray to determine the amino acids that are going to be present in this structure. So in order to determine that, you need to have a genetic code table. And in the genetic code table, you will see the different codons and their corresponding amino acids. So AUG, and I'm going to give the 3-letter code for the amino acids. AUG is methionine. ACU is threonine. AGC is serine. UGG is tryptophan. GGG is glycine. Then UAU and UAC are both tyrosines. UUU is phenylalanine. And UAG stands for a stop codon. A stop codon is going to be a place where we have the release factor bind. So because, let me just take a note here. No tRNA because of site for release factor. So remember, stop codons do not encode for an amino acid. That is a place for the release factor, so there's also no tRNA or anticodon for it. A release factor is a completely different molecule. So for the DNA given on number 2, that is specifically the template strand. So I'm going to be writing its complementary strand, which is going to be the coding. And remember that that coding is going to be running in the 5' to 3' direction. And then the template is the one that runs 3' to 5'. So the coding strand for the strand of DNA given is going to be ATG, GCG, AGG, CGG, CAG, CTG, TTA, TGG, TGA. So understand that once we have this, then it's fairly easy to write the mRNA, because the mRNA is going to be very similar in sequence to the coding strand. It's just that the T's are going to replace by U's. Remember that the reason why that is because both are going to be synthesized from the template strand by different enzymes, but that's the way it goes. So when it comes to the mRNA, this is going to be AUG, GCG, AGG, CGG, CAG, CUG, UUA, UGG, UGA. When it comes to the tRNA, remember I already explained how it is complemented to the mRNA. So the tRNAs that are going to be bringing those amino acids for number 2 are going to be UAC, CGC, UCC, GCC, GUC, GAC, AAU, ACC, and we are not going to have a tRNA for that last one, because we're going to see that when we have written the amino acids, remember, we are going to be paying attention to the genetic code. UGA actually encodes for a stop codon, so that's why there's no tRNA. The amino acids that are brought by those tRNAs are methionine, alanine, followed by arginine, glutamine, leucine, tryptophan. And in the last problem, we are given the sequences of amino acids. Remember that there's going to have different answers. I am just selecting codons. So if you wrote a different codon that corresponds to the amino acid, understand that you may be correct, as long as you use the genetic code table. So if you're given the sequences of amino acid, the easier thing to write down is going to be the mRNA. So methionine is always AUG. Arginine, one of the codons for it is going to be AGA. For glycine, one of the codons for it is GGU. For phenylalanine, we have UUU. For methionine, it is always AUG. Valine is GUU. Glycine, GGC. In the stop codon, I just selected one of the 3 choices. I'm going to write it as UAA. So once I know my mRNA, one of the things that I can do is just write the tRNAs that are going to be bringing the amino acids that I have written already. And the tRNAs are going to be UAC, UCU, CCA, AAA, AAA, UAC, CAA, CCG, and remember that there's no tRNA for your stop codon. Then, since we know the mRNA sequence, then out of the two strands, it will be easier to write the coding. And again, that one runs in the 5' to 3' direction. And the coding strand is going to look the following. It's going to be ATG, AGA, GGT, TTT, TTT, ATG, GTT, and GGC, and the last one is going to be TAA. If I write in the template strand, it's going to be complementary to the coding. And remember that the template strand is going to run in the 3' to 5' direction. And the template strand for this is going to be TAC, TCT, CCA, AAA, TAC, CAA, CGG, ATT. In the next page, we are going to be labeling the following figure, which has the different parts of the cell. This is going to be representing a eukaryotic cell. So I'm going to write down the letters because we need to be labeling the parts from A through J. So we have A, B, C, D, E, F, G, H, I, and then J. Let me do that. Let me write them a little bit more up. This is A, B, C, D, E, F, G, H, I, J. So, A is representing a molecule of DNA. B is representing a molecule of mRNA. C is representing your tRNA with an amino acid. Letter D represents a polypeptide. Letter E is representing the anticodon, because observe that anticodon is part of the tRNA. F is representing the codon, because we're looking at a 3-letter code in the mRNA. Part G is that shadow that we have in the back, and that shadow is going to be the ribosome. Letter H is the nucleus of the cell. That's how we know that this is a eukaryote. Letter I is going to represent the process of transcription. And lastly, letter J represents the process of translation. Notice that the process of transcription in a eukaryotic cell happens in the nucleus, while the process of translation is going to be happening in the cytoplasm.